Greetings. I am Captain Tsirk. In this video I will discuss the Human Genome Project, which is an international scientific research project with a primary goal of determining the sequence of chemical base pairs which make up DNA, and of identifying and mapping the approximately 20,000 to 25,000 genes of the human genome from both a physical and functional standpoint. The project's goals were to identify all the approximately 20,000 to 25,000 genes in human DNA, determine the sequences of the 3 billion chemical base pairs that make up human DNA, store this information in databases, improve tools for data analysis, transfer related technologies to the private sector, and address the ethical, legal, and social issues that may arise from the project. The Human Genome Project was one of the great feats of exploration in history. It was an inward voyage of discovery rather than an outward exploration of the planet or the cosmos, an international research effort to sequence and map all of the genes, together known as the genome, of members of our species Homo sapiens. Completed in April 2003, the Human Genome Project gave us the ability to, for the first time, to read nature's complete genetic blueprint for building a human being. While the objective of the Human Genome Project is to understand the genetic makeup of the human species, the project has also focused on several other non-human organisms such as E. coli, the fruit fly, and the laboratory mouse. It remains one of the largest single investigative projects in modern science. The genome of any given individual, except for identical twins and cloned organisms, is unique. Mapping the human genome involves sequencing multiple variations of each gene. The project did not study the entire DNA found in human cells. Some heterochromatic areas, about 8 of the total genome, remain unsequenced. The best estimates of total genome size indicate that about 92.3% of the genome has been completed, and it is likely that the centromeres and telomeres will remain unsequenced until new technology is developed that facilitates their sequencing. Most of the remaining DNA is highly repetitive and unlikely to contain genes, but it cannot be truly known until it is entirely sequenced. Understanding the functions of all the genes and their regulation is far from complete. The roles of junk DNA, the evolution of the genome, the differences between individuals, and many other questions are still the subject of intense interest by laboratories all over the world. Key findings of the Human Genome Project are that there are approximately 30,000 genes in human beings, the same range as in mice and roundworms. Understanding how these genes express themselves will provide clues to how diseases are caused. Also, between 1.1 to 1.4% of the genome's sequence codes for proteins. The human genome has significantly more segmental duplications or identical, repeated sections of DNA than other mammalian genomes. These sections may underlie the creation of new primate-specific genes. At the time when the draft sequence was published, less than 7% of protein families appeared to be vertebrate-specific. How this was accomplished was the genome was first broken into relatively large chunks, which were then mapped to chromosomes before being selected for sequencing. If the sequence obtained was to be stored in book form, and if each page contained 1,000 base pairs recorded and each book contained 1,000 pages, then 3,300 such books would be needed in order to store the complete genome. However, if expressed in units of computer data storage, 3.3 billion base pairs recorded at 2 bits per pair would equal 786 megabytes of raw data. This is comparable to a fully data-loaded CD. Advantages of Human Genome Project include knowledge of the effects of variation of DNA among individuals, which can revolutionize the ways to diagnose, treat, and even prevent a number of diseases that affects the human beings. It also provides clues to the understanding of human biology. Thank you for watching my video on the Human Genome Project. Please feel free to leave any comments or questions. Goodbye.